Okay, can you guys hear me okay? Is the microphone working? Okay. So welcome to another beautiful fall day in Flagstaff. For those of you not from around here, the rain today, I guess at this time in September is sort of unusual. Anyway, it's sort of funny, Scott and I started off our talks with fairly similar photos, but there is a difference. I think, Scott, yours was a composite from satellite remote sensing, right? Your Earth.
It was an image of Earthrise over the moon.
But was it a real picture, or was it a okay? It wasn't the Modus composite.
No.
Okay. Well, anyway, so we both then have Apollo pictures to start off our talks. I'm going to come back to this picture a little bit later, but it's 45 years since Apollo 17 astronauts took this picture from space. And this picture is unique in that it's one of the few human-recorded images that shows a fully illuminated Earth. And Apollo 17 was the last manned lunar mission, and in the decades since, no human has been back in the same position to take a picture that shows the whole Earth disk. And I've read before that this picture is the single most reproduced photograph in history. And I just want to show this to get you thinking about the power of photographs, particularly as a record of a particular scene at a particular point in time. And I am not advancing. Okay, so today I'm going to talk about how with the PhenoCam network we're using images from networked digital cameras to track vegetation and phenology in a diverse array of ecosystems across North America. And I'll talk about what this is telling us about connections between phenology, ecosystem processes, and climate change. And I want to acknowledge the contributions of my collaborators to this work, and also funding we've received from various federal agencies. And last but not least, I want to thank all our collaborators on the PhenoCam project. This includes site PIs and technicians who put our cameras out at their sites and maintain them. And without their support of PhenoCam, a lot of what I'm talking about today would not really be possible. And so just to be clear what we're talking about, I want to distinguish between phrenology, which is basically the study of bumps on the head, and I have to admit this slide is always good for at least a few nervous giggles. So we're not talking about phrenology, that lecture's on the other side of campus, but phenology, which is really the study of seasonality and seasonal rhythms, or the dictionary definition is the scientific study of periodic biological phenomena such as flowering, breeding, and migration in relation to climatic conditions. And the pictures here all show classic examples of different types of phenology, from spring emergence and flowering by plants, insect metamorphoses, migration of mammals and birds, and then also ripening of crops and fruit, and color change and senescence in autumn. And unfortunately, I realized when Lauren was talking about this g2p2pop RCN and sort of focusing on vertebrates, this is the end of what I've got for vertebrates, so bear with me. But the point is that phenology is really all around us all the time. As we cycle through the seasons, new phenological events are constantly occurring. Now, phenology is inherently of interest to biologists because it represents this intersection point between organisms and their growth environment. But what I think is fascinating is that phenology is also just inherently of interest to the general public. People seem to be inherently connected to and fascinated by the passing of the seasons and the way in which symbols of that passing are manifest. And so phenology has been lyrically referred to as the pulse of our planet and the rhythm of the seasons. And Scott showed some really nice NASA remote sensing videos, and this is an older, lower resolution video, but this is also produced from NASA satellite remote sensing. And it shows the annual cycles of vegetation development and senescence around the globe. And I think you get a real sense of that pulsing and the rhythm of the seasons from this video. Okay, so over the last couple of decades, there's been a real surge of interest in the field of phenology, and it's increasingly recognized as really an essential component of cutting-edge global change science. And going back to this picture, this is the famous blue marble photograph. And the Earth has really changed a lot since Apollo 17 astronauts took this picture on December 5th, 1972. Sorry, December 7th. So we're seeing a lot of changes in the Earth's atmosphere. Scott talked about these. We're seeing rising concentrations of the greenhouse gases, carbon dioxide and methane. And these atmospheric changes are then driving recent changes in the climate of our planet, including warmer temperatures and altered precipitation regimes. And these changes in climate are then affecting how ecosystems work. And so phenology is getting a lot of attention these days because it's really been shown to be a leading indicator of the biological impacts of climate change. So there's substantial evidence in the literature for the advancement of spring phenological events, such as leaf out and flowering. These are moving forward at a rate of about 2 to 5 days per decade since the 1970s. So think, up to a couple of weeks over since I was born at least. Similarly, there's many examples of autumn phenological events, such as leaf coloring and autumn senescence. These are all later. And so these studies show quite conclusively that the growing season is changing, and studies like this have been widely reported not only in the scientific literature, but also in the popular press. And this gets back to the idea, as mentioned earlier, that humans are just fascinated by the passing of the seasons. And I think that phenology is really important as a biological indicator of climate change because it's a way of, very tangible way of conveying to the public what these impacts of warmer temperatures and shifts in precipitation might be. And then this is going back, it's kind of ancient history now, but in 2007 in the IPCC's fourth assessment report, they concluded that phenology is perhaps the simplest process in which to track changes in the ecology of species in response to climate change. So there is a lot of surge of interest in phenology in the last few decades. And these shifts in phenology are important for a number of reasons that cut across different scientific disciplines. So, for example, in terms of ecological interactions, shifts in phenology affect herbivory, competition, pollination, and dispersal. In terms of ecosystem services and climate system feedbacks, shifts in phenology affect the albedo, or the reflectivity of the surface. They affect carbon and water fluxes, and also the surface energy budget. And then from the perspective of human society, shifts in phenology are important for public health issues, including allergens and disease vectors, but also agriculture, so planting and harvesting schedules, irrigation schedules, and so forth. And they're also really important for tourism, so wildlife viewing, bird watching, leaf peeping, wildflowers, and so on. So there's all sorts of reasons that shifts in phenology are important from the perspective of both science and human society. So I'm going to talk a little bit now about some of the specifics of what we're doing with the PhenoCam network and how this is informing our understanding of the impacts of climate change on phenology and following on that on terrestrial ecosystems more generally. So PhenoCam does what we refer to as near-surface remote sensing. We're using imagery from network digital cameras. The cameras sort of look like this. They're about the size of my fist. We're continuously monitoring plant canopies with the imagery from these canopies. Imagery from these cameras, sorry. Images are recorded every 30 minutes from sunrise to sunset, 365 days a year. So these guys are basically working for us non-stop. And the scale of observations, Scott talked a lot about scale in his talk and the whole NASA scaling strategy from leaves to ecosystems to biomes. With PhenoCam, the scale of the observations we're getting from these camera images is conveniently intermediate between what's seen on the ground at sort of the level of individual organisms and what's seen by satellites at a much coarser spatial and temporal resolution. And that gives us observations that can help to inform our understanding of tower-based flux measurements, stand-level forest inventories, and small watershed measurements. So I'm just going to run through some PhenoCam imagery for Harvard Forest. Harvard Forest is located in central Massachusetts. The canopy there is dominated by oak and red maple. And this shows a picture in late April when the canopy, the snows have melted, but the canopy is still in a dormant state. And if we fast forward a few weeks into early May, you see the leaves are just beginning to come out. The canopy is this brilliant sort of bright green color. We go into late June. The foliage is maturing, it's expanding, it's thickening, the pigments are developing. Leaf area index peaks here about the end of June, which is what you're seeing here. By August, we're beginning to see an overall yellowing of the canopy as a whole. You can see some trees here are beginning to show their autumn colors. And then by mid-October, the canopy is this really nice mix of golds and reds and browns that's really typical of autumn in southern New England. Can you guys hear me okay if I stand back a little bit farther? Okay. And then by December, the leaves have been shed and the canopy is back in this dormant state that won't be broken until just before the start of the following growing season. So the canopy images, sort of like that blue marble photograph, they're a permanent record of what the canopy looked like at a particular point in time. And we can look at the images and visually estimate the date when leaves emerged in spring, or when they started to turn color in fall, or when the canopy had lost all its leaves at the end of fall. But we can also directly extract quantitative data from these images. And the reason for this, or what makes this possible, is that digital camera images are based on the red, green, blue additive color model, which basically says that we can reproduce any color as perceived by the human eye through some combination of these three primary colors. And what that means is we can take a PhenoCam image like this is one of the Harvard Forest pictures, and we can split it into these three component layers, the red channel, the green channel, and the blue channel. And then for a particular region within each image, which we can just define based on the question we're interested in studying, but for that particular region, we can extract quantitative data from the image that describes in numerical terms the color of that region, and that gives us what we call this red, green, blue triplet. It's basically the brightness of each of those three color channels averaged across the region of interest. And so what we're starting to do then is take an image from just a standard digital camera and think of it more like a satellite remote sensing image with multiple channels. And we can take our quantitative data on the brightness of the red, green, and blue channels and convert that to indices that sort of simplify going from 3 dimensions here to a single dimension, simplify the change in the vegetation over time. So this is our Greenness Index. It's basically just the relative brightness of the green channel normalized the sum of the brightnesses of the red, green, and blue channels. And when we calculate that index for a particular region within the image across multiple years of imagery for Harvard Forest, this is what we get. So this is showing 6 years of canopy greenness data derived from that PhenoCam imagery. And you can see here that greenness is low in the wintertime. It rises steeply in spring as those leaves emerge and they expand and mature. It drops off gradually over the summer as that canopy yellows slightly. Drops off rapidly in fall as leaves turn to yellows or to oranges and golds and browns, and drops down to a minimum as those leaves are shed from the canopy. And then the cycle repeats itself in the following years. Now with a time series like this, we can use various methods, curve fitting methods and so forth, to extract inflection points from the data, and those inflection points then correspond very nicely to on-the-ground observations of leaf emergence, peak canopy development, onset of leaf coloration, and completion of leaf fall, as recorded on the ground by a human observer who's actually out looking at the trees. So the point is that this PhenoCam approach gives us data automatically and at very high temporal resolution but that is comparable to and consistent with on-the-ground observations made by a human observer. And then uniquely, because these are images, and because they're very high-resolution images, we can select what part of the image it is that we're looking at, whether we want to average or aggregate across all of the multiple tree crowns in the field of view of this camera, or whether we want to split out the different species and different individuals and look at them separately. Now, the same approach is also able to track greenness, seasonal changes in greenness in other ecosystem types. So for almost 8 years now, we've had a camera looking out at this mixed C3, C4 grassland. It's located at about 3,000 feet on the western side of Mauna Kea on the Big Island of Hawaii. And seasonality here is not driven by winter, spring, autumn, winter kind of seasonal transitions and cold temperatures versus warm temperatures, but much more by the timing and magnitude of precipitation events. And these tend to be much less regular in time than the seasonality of deciduous forests in the northeastern United States. You can see here, this is our Greenness Index derived from 3 years of imagery from this camera. And in some years we get 1 or 2 green up cycles. In some years we get 3 or 4 green up cycles, and the timing and duration of these cycles varies just depending on those precipitation inputs. But again, we're able to quantify that automatically at a very high temporal resolution from this camera imagery. And you can see here, so if you're looking at this movie, you can see we get very green at times, and now we're very brown, and now we're very green. And that is then represented by this Greenness Index. So what about evergreen vegetation? You know, whether ponderosas around here or evergreen forests in the tropics. Turns out that some evergreen forests are much more seasonal than others, and the amount of seasonality appears to really depend on the amount of cold temperature that's received. And we can again characterize that from the camera imagery. So this is showing Niwot Ridge, Colorado. This is just north of the town of Nederland, if you're familiar with Ned. Niwot Ridge is a high elevation conifer forest dominated by spruce, fir, and pine species. And you can see here, we still see, even though we think of it as an evergreen forest and always green, well, there's times where it's a lot more green in the peak of the growing season, times it's a lot less green in the wintertime. And we can quantify that from the camera imagery. And it's, you know, this strong seasonality that this evergreen forest, which always has foliage, that it exhibits. And we've done chemical analyses looking at the leaf pigments in the foliage and how that changes seasonally. What we can say is that the seasonality in this forest, unlike the deciduous forest in the northeast, which is, the seasonality there is driven by the production of new foliage and the shedding of foliage, existing foliage in the autumn. Here the seasonality is driven by biochemical changes within existing foliage, so changes in the amounts of the leaf pigments, chlorophyll, carotenoids, and xanthophyll cycle pigments, and how those vary seasonally. That's what's driving those changes in greenness. And this is just showing here a picture from July on the left and January on the right. You can see just visually how much greener that foliage is in the summertime than in the winter, when it takes on more of a rusty, greenish brown kind of color. And if you look at the ponderosa pines around here in wintertime, you'll see the same kind of thing. They just don't sort of pop in the same way. They don't have that bright green that they do at the end of the monsoon. So in other evergreen ecosystems, this seasonality is not so pronounced. So this is 2 years of data from a camera that we've got set up again on the Big Island of Hawaii, but this time near Thurston Lava Tubes in Volcanoes National Park. And this is there's a variety of evergreen tropical species, Metarosideros, Ilex, and Chibodium. And the y-axis on this plot is the same as in the Niwot plot, but you can see, although there is some seasonal variation, it's nowhere near as pronounced as at Niwot Ridge. So this gets the idea that the more seasonality there is in climate, the more seasonality we see in greenness in these evergreen forests. I don't know how many people in the room are remote sensing folks other than the group from Scott's lab that's here, but you're probably wondering about whether we can get information about near-infrared wavelengths from our camera imagery. And in fact, we have figured out how to turn our RGB or red, green, blue cameras into relatively inexpensive 4-channel imaging sensors that have this near-infrared band. And what makes this possible is the fact that the CMOS imaging sensor that is common in most digital cameras is in fact sensitive to wavelengths beyond what the human eye can see, so beyond near-infrared wavelengths beyond 700 nanometers. Now these wavelengths are typically blocked by a filter inside the camera, the idea being if you want your image to represent what the eye sees, you don't want those wavelengths that the eye can't see to be affecting the image. In our cameras, which were designed for security applications, we can actually trigger that filter through software. And what we do is we take sequential images, back-to-back pictures, one with the filter in place, which is a visible wavelength-only picture, and we follow that up with the filter removed, and that's a visible plus near-infrared image. And then we difference those two images to pull out the near-infrared band. And this has several neat applications. You can see false color images on the bottom here. This is using sort of the old-fashioned satellite kind of mapping, which puts the infrared onto the, onto the red band, with this representation of it that includes the near-infrared wavelength information, vegetation comes out as this luminous pink. So it's called false color because it's still a color image, but it's different from what your eyes would normally see. So we can do that with the near-infrared channel, or we can calculate an index, which we call camera NDVI, that is analogous and quite similar to the normalized difference vegetation index that's commonly used in satellite remote sensing. So the first PhenoCam was installed at Bartlett Experimental Forest in the White Mountains of New Hampshire in 2005. Originally we put it up just so we had pictures of our canopy. We could use them in talks and stuff. We didn't think we'd get phenological information. But we did quickly found out that we could get quantitative data using this red, green, blue extraction that I talked about. And by 2008, I had a small amount of funding to start a regional network focused on the phenology of forests in the northeastern United States and adjacent Canada. And that was really just the start of things. I never had imagined that 12 years later I'd still be doing this. But over the last 10 years, PhenoCam has really blossomed into this continental scale phenological observatory that is now. Currently we have over 500 cameras contributing imagery to the PhenoCam data set. Most of these are located within the United States. We have cameras from Alaska to Florida and from Hawaii to Maine. But we also have cameras in Canada, in Panama, several countries in Europe as well. And the imagery and data from these cameras is all publicly available in near real time through our project webpage, which for historical reasons, because I started this when I was originally at UNH, and all the cameras are now sending images, have always been sending images directly to the server there. We just left the server at UNH, but that's the address. You can just Google it and you'll get directed there. So it's just showing sample images from a variety of different ecosystem types. We've got evergreen and deciduous forests, grasslands and prairies, savanna and shrublands, as well as a number of agricultural sites. And ultimately our goal is to have a comprehensive sampling of all the diverse ecosystem types in North America. So I want to talk now about some of the things we're learning from these data, but I also get tired of standing at the front of the room and just talking. Does anyone have any questions on the first part of my talk? At high elevations in the Appalachians, and then the PhenoCam models not doing so well here down in the deep south. But overall, we got a low bias and a median root mean squared error of about 5 days from both modeling approaches, which is a huge improvement over existing methods that are used in current generation land surface models. And so previous studies have failed to generalize these generalized phenological models that failed to develop phenological models that generalize well in time and space. But here, using these PhenoCam data from across a range of different sites, we actually did come up with a very simple model structure that does generalize pretty well across the entire eastern deciduous forest, and that has the advantage of not requiring species level information. And so this shows really the potential for developing improved phenological models to be included in more complex land surface models. So as a next step, we took Eli's PhenoCam model, the species ignorant model, and put it into CLM, the community land model. CLM is one of these large Earth system models that is run forward for 100 years to predict how the climate's going to change. And we used this new model as a replacement for CLM's standard winter deciduous phenology model. And this is work that was led by my former postdoc Min Chen. It was published a couple of years ago in Global Change Biology. So here the old model is the existing model in CLM, and the new model is the species ignorant PhenoCam model. And in terms of model predictions, you can see that the two models sort of have similar geographic patterns. There's a gradient of earlier spring onset in the southern latitudes, and later spring onset at more northerly latitudes. Globally, if you average across the whole deciduous forest, the two models predict about the same date of spring onset under current climate conditions of May 1st. But if you start to really dig in, you'll see that the differences between the models are greatest at the highest latitudes and the lowest latitudes. You can see some subtle differences in the color gradient here versus here, for example. And when we validated the models globally, again using MODIS data, the old model has a root mean squared error of about 7 days, and the new model manages to cut that in half to about 4 days. So again, a substantial improvement with this new species ignorant model. And this is largely because of particularly poor performance of the existing model in some areas, so in the southeastern United States, in Eastern Europe, and in Southeast Asia, where the new model here is doing much better. So we then were interested in looking at, well, what does this imply for future changes in phenology? So we ran the two phenology models forward to 2100 using climate projections, and we did this for all of the northern hemisphere deciduous forests. And you're seeing here maps of the shifts in deciduous forest spring budburst by the end of the century, with green indicating earlier spring onset and red indicating later spring onset. And here again, the old model is the existing phenology model in CLM, and the new model is this species ignorant PhenoCam model. And what's interesting is that the existing model, the old model, predicts relatively little change in spring budburst across the global deciduous forest biome by the end of the century. And in some areas, particularly the southeastern United States and southern Asia, it's predicting that spring is actually going to occur later. Those leaves are going to be coming out later than they are presently. And these trends towards later spring that this model predicts are kind of in direct conflict there between the two models. And this is for the RCP 8.5 climate scenario, so a fairly aggressive climate change scenario. Now, one thing with CLM is that it's fully simulating ecosystem processes on the land surface. And what that means is that in doing all this modeling, we can satellite land cover classification. This is just showing across that range for 10 different climate models, each graph representing a different model, the projection of future warming by the end of the century from that climate model. What you can basically see is the models are in agreement that it's going to get warmer by 2100, which may not be much of a surprise. The models are less in agreement, though, on precipitation patterns, precipitation changes over the coming century. But there's generally a consensus towards somewhat wetter conditions in the northern part of the Western biome and somewhat drier conditions, but less precipitation in the southern grasslands. But overall, the precipitation changes are expected to be relatively small. But when you take increases in temperature and put that together with small changes in precipitation, what you do find is that there is a projection of a consistent increase in what's known as the aridity index across the grassland biome. And aridity is just it's the ratio of mean annual precipitation to mean annual potential evapotranspiration. And these changes are really mostly driven by that rising temperature, negligible changes in precipitation. But what we want to do is use a model-based approach to look at how changes in temperature, precipitation, and aridity, how these environmental changes might be expected to influence grassland phenology and productivity. And our model couples plant growth with soil hydrology so that we can capture that pulse response flux dynamics that are really characteristic of grassland ecosystems. And this is just showing the overall sort of model structure very schematically. We're using temperature, soil radiation, and precipitation as environmental factors that drive the model. At each time step, we're tracking two vegetation states. So vegetation cover as a measure of carbon, and soil water as our sort of water state. And then we're modeling the flows or fluxes of both carbon and water at each time step. So the senescence and growth of vegetation influencing the vegetation cover, and the evapotranspiration and runoff influencing soil water. And what you can see here from the schematic is that the coupling between carbon side processes and water processes are explicitly represented here. So soil water influences vegetation growth and senescence, these arrows, and then vegetation cover influences evapotranspiration. So we use this model to look at how the grassland biome might respond to these future climate changes. To calibrate the model, we used canopy greenness from PhenoCam imagery as a proxy for vegetation fractional cover. And we used more than 30 site years of data from 14 different PhenoCam sites to train the model. Now, first of all, what we did was we did a one-out cross-validation, so we used data from 13 sites to train the model, to calibrate the model parameters, and we tested it against the 14th, and we repeated that 14 times so that each site was independently tested. And then once we were convinced that the model was working pretty well, we estimated a single set of model parameters that we applied across all of grassland sites. And this is just showing the cross-validation for a C3, C4 site in Oklahoma. The estimates of fractional cover from the PhenoCam imagery are shown by the dots. The red line is the model simulated trajectory of fractional cover, and the blue lines are precipitation inputs, which you can see really are driving the seasonality here. And on the right, this is just reprojecting this into sort of two-dimensional scatter plot space. But the take-home message here is that the model is doing a pretty good job at representing the seasonal dynamics of this site, and we had similar results for other sites. We then took the model, ran it forward for 10-kilometer grid cells to the year 2100, using the output from each of the 10 different climate models that I showed earlier. So we had 10 different sets of model runs, one for each model. And basically the take-home message here is that we're going to see an overall increase in productivity of about 10 to 20%. It's in this kind of range here across the grassland biome. The response is stronger in the north. These are dark greens, weaker in the south. These are lighter greens and yellows. The differences between the model runs were generally related to the model's projections of changes in precipitation. But this shaded area indicates where across those 10 models we had a statistically significant increase across all model runs in productivity. And a sort of interesting finding to emerge from this modeling was that we were predicting that the grasslands are going to respond to future climate change by shifting their seasons. And these plots on the right, these are showing the changes in seasonality of vegetation cover between 2010 and 2100. So the difference between the seasonal cycle in 2100 and what it is right now under sort of current climate conditions. And generally what we see is earlier onset and enhanced vegetation productivity in spring, and delayed senescence and enhanced productivity in fall. That is offset to some degree by reduced productivity in summer resulting from summer drought. Now, for the northern grasslands in our study, kind of about 80% of the study area shown here by this darker green, our modeling suggests that these increases in spring and autumn more than offset that summer decline related to drought. And so we'll see a net increase in productivity up in this part of the grassland biome. For the more southern, more water-limited sites, yes, you'll see an increase in spring, and you may see a bit of an increase in fall, but that is offset by a more severe reduction associated with summer drought. So in the southern range, little or no change in overall productivity. Now, one thing to point out here is that some other studies have recently shown that they've associated climate change with increases in agricultural productivity, but at the cost that this enhanced quantity of agricultural production is accompanied by the reduced quality of that production. And so less nutritionally, less nutritious crops and grains, for example. And so I don't want to make it sound like we're saying that climate change and enhanced productivity is necessarily a good thing, because there may be other effects that we didn't quantify from our model. But anyway, the take-home message of the study is there in the title, that we expect that the productivity of these grasslands to be increased under future climate change in spite of this increasing aridity. We predict that earlier spring and delayed senescence more than compensate for summer drought effects, and this results in overall enhancements in grassland productivity. We do expect that changes in seasonality will present some management challenges as we just sort of move beyond the range of what's been historically experienced. So, grazing, planting and harvesting schedules, irrigation, and so forth, these are all going to be unlike maybe what farmers are used to in the past. And I think it's pretty clear that these future shifts in phenology and growth are then going to influence ecosystem services that do have real economic value to human societies. So I've got one more part to talk about. Any questions on the grasslands? Yeah.
Do you include any kind of herbivore grazing in those models, and any feedbacks that occur?
No, that's a tricky thing. Definitely, yeah, we don't. We did, we did do some independent validation of the model predictions using cuttings from an experimental site in Kansas, but that is not a grazed site, so I can't really say how grazing would play into things, but grazing isn't represented in a lot of these big scale models anyway. It's sort of like, yeah, it's an obvious component that's missing. Okay, so finally I'm just going to talk about how we're using PhenoCam data to track vegetation phenology in an ecosystem warming experiment. And we're partnering with scientists from the Department of Energy's Oak Ridge National Lab, who've set up what is called the spruce experiment in a boreal peatland forest in the north central United States. And this is a 10-year experiment. These chambers, there's 10 of them. They're about 12 meters across and 8 meters high, and they're being used to apply a range of experimental temperature and carbon dioxide treatments to mature and intact patches of peatland vegetation. And this just shows the experiment from the air. This is the peatland bog where this experiment is taking place. You can see the chambers scattered across the bog, and this evergreen-dominated bog is surrounded by deciduous forests in the uplands. Now, overall, the experimental design is intended to simulate environmental conditions that may be experienced by the end of the century. And there's 10 chambers in total, as I said, 5 levels of warming from 0 to plus 9 degrees Celsius above ambient, which is absolutely a huge increase if you think about it. And I'll put that in perspective in the next slide. And those warming treatments are then crossed with 2 carbon dioxide treatments, so ambient carbon dioxide at 400 parts per million and elevated carbon dioxide at 800 parts per million. The warming treatments were turned on 3 years ago in August of 2015, and the CO2 enrichment was turned on in June of 2016. So we're 3 years into what's supposed to be a 10-year experiment. Just to put the 9-degree treatments in context, anyone have any idea where, if you were to move spruce south, how far south would you have to go to simulate 9 degrees increase? Like, maybe South Dakota? Nebraska? Any guesses?
Texas.
So was that?
Texas.
Texas, not quite Texas, but Tulsa, Oklahoma has a very similar climate. It is just about 9 degrees warmer and has pretty similar precipitation to the outside of the chambers at the spruce site. So if you think about what that, that's a pretty big change. So we installed digital cameras with these PhenoCams in each of the spruce chambers in August 2015. This is the single picture I've ever taken using a selfie stick. The cameras are mounted at a height of 6 meters on the south wall of each chamber, so they're a very consistent mounting point. The cameras then have a view of the interior, full interior of each chamber. And the cameras are recording images every half hour and uploading them to our PhenoCam project webpage, which, like all these cameras, allows you to check what's happening at the parasite from anywhere in the world at any time. And I was talking earlier about this power photographs as this permanent record, and I think one neat thing about using cameras in this experiment is that the imagery gives us a permanent record of what each chamber looked like at a particular point in time, every half hour over the course of the experiment, over its 10-year course. And so this is just looking New Year's Day 2016. This really shows the effectiveness of the experiment. This is chamber 19. This is an unheated enclosure. So at -5°C in this chamber, there's snow on the ground. And this is chamber 17, a +9 plot. It's about 5 degrees above freezing, 5°C above freezing, and there's no snow here. And so these images allow us to compare the different plots and to track the experiment over its 10-year course. But also we're interested in using the camera data to follow how the experimental treatments are influencing the phenological patterns. And there's 3 different competing hypotheses which we're explicitly interested in testing here. So, if phenological transitions are controlled strictly by temperature, illustrated here on the top, we should see a strong and direct relationship between the air temperature in the chamber, with these being the unheated plots, these being the +9 plots, and the phenological transition date that we infer from the camera imagery. It's also been suggested that some phenological events are controlled by photoperiod or day length, and this is particularly the case with autumn senescence and the shedding of leaves in fall. And if that's the case, that phrenology controls some phenological events, then we should see no relationship or a flat relationship between temperature and the phenological transition date, because the day length is the same in each of these chambers regardless of the amount of warming. And then it's also been suggested that warming will only have an effect up to a certain point, and then there will be this photoperiod constraint that kicks in and limits additional phenological response to temperature. If that's the case, we might expect this sort of kinked relationship where up to a certain amount of warming we see an effect on phenology, and then beyond this temperature threshold T-star value, it's flat as photoperiod prevents the vegetation from phenologically shifting further. And we're going to test for evidence of that breakpoint temperature T-star. So this is a view inside chamber 6. This is one of the unheated enclosures. And I talked about how we can look at different organisms within a camera field of view, and we're doing that here. So we subdivide our image into 3 different vegetation types. These yellow trees, these are larch larix, a deciduous conifer, so it sheds all its foliage in fall. We also have black spruce, an evergreen conifer, which is the most abundant tree species. And then we have a mixed shrub layer on the surface of the bog, which is dominated by Labrador tea and leatherleaf. And we look at each of these 3 vegetation types separately. And what I'm showing here, 3 vegetation types, so larch, picea or spruce, and the mixed shrub layer. You see that autumn senescence is delayed by warming, so this is the plot temperature here, and the date of autumn senescence on this axis, and you can see in all cases we're delaying senescence with those warmer temperatures. And on the bottom here you can see, again, across those 3 vegetation types, that in all cases spring green up is advanced by warming. So there's some differences in the temperature sensitivity across these vegetation types. You see differences in the slope, but overall senescence is delayed by about 3 days per degree of warming across the treatments, and green up is advanced by about 2 days per degree across the treatments. And what this means is that across the 9-degree temperature range that we have here, there's a difference of more than 6 weeks in the length of the active season between the unheated and the most significantly warmed plots. Now, in almost all cases, in all cases really, the response to warming is quite linear. The fact that the slopes on all these relationships are non-zero allows us to reject that photoperiod only observer was looking at the phenology of these species in response to warming. And in all cases, the response to warming, the trends were highly significant for all species and all different phenophases, with a mean change of about 3 days per degree, quite similar to what we got from the camera data. And again, in these ground observations, very little support for that breakpoint model. So then, last thing to talk about is just how we had an extreme weather event that really showed dramatically how the warming treatments are triggering physiological changes, specifically, in this case, advancing the dehardening of tissues and opening them up to the potential for frost damage in springtime. And so what happened was we had a very warm period of weather in March. It was the second or third warmest March on record for the area, and that was followed by extremely cold weather in early April. So in the warmest +9 chambers, we had accumulated at almost 8 times as many degree days above 0 degrees Celsius in these warm chambers as outside of the plots. So there's a combination of both the warming treatments and the warm weather. Because of the warm weather and the warm treatments, it had been more than a month since these 2 chambers dropped, since the temperature inside those chambers even momentarily dropped below -2.5 degrees Celsius, and that is seen as a sort of critical temperature beyond which freezing damage may occur in unhardened tissues. And then on the morning of April 9th, this blast of Arctic cold air came through. Outside of the chambers, these ambient plots, the temperatures dropped to almost -16 Celsius, and even in the +9 chambers, it dropped to -3 to -4.5 degrees Celsius. And even particularly in plot 17, the spruce in that plot suffered almost immediate and extremely severe frost damage, which we can then identify quite nicely from the camera imagery. So this is plot 17. That morning after the frost event hit, everything looks okay, but 10 days later, you can see how that spruce tree in the middle, it's changed color and is about to, it will over the following month lose most of its foliage. This larch down here, you can see that it's just beginning to leaf out in this picture. 10 days later, it had some damaged foliage, but it put out a new flush of needles. And so it was not particularly harmed by the freezing event in the same way that the spruce here was, or that spruce back there. Now, what was really interesting was that the damage in the other chambers was negligible or non-existent, even though those chambers experienced much cooler temperatures because they were heated less. So in those chambers, the vegetation still retained sufficient frost hardiness to tolerate those lower temperatures. And for this tree in the middle, this next plot just shows about a month later what the branches look like after that dead foliage was shed. You can see they retained a few green needles from the previous growing season at their branch tips. These buds here, they were still okay. They burst and put out a new flush of foliage sort of on schedule about 6 weeks later. But these trees went into the 2016 growing season with having lost a large amount of foliage and significant amounts of leaf nitrogen that was in that frost-damaged foliage and was not retranslocated. Okay, so we're tracking phenological responses in this warming experiment using PhenoCam imagery, and we're seeing that warmer temperatures are extending the growing season at both the start and end of the growing season in this boreal black spruce bog. So far, we've got little or no evidence for photoperiod control of phenology, as the response to these phenological transitions to temperatures is generally linear and significantly different from 0. And then we saw in the warmest plots that early loss of winter hardening resulted in severe frost damage. And this is actually consistent with ideas that have been put forward in the literature that warmer temperatures in the future may enhance the risk of frost damage. Frost damage may become more common in a warmer world. And then over the long term, one thing that I think we'll see is shifts in vegetation phenology as an indirect effect of the warming as we start to see shifts in community composition within the chambers, particularly the +9 chambers. And the neat thing is we'll be able to track those changes in community structure from the camera imagery. Okay, so I mentioned the data are all publicly available through the project webpage. We've also recently put together and documented a curated data set